Hi, everyone, and welcome to day two of this virtual Firefly Symposium, an event focused on firefly science, conservation, and education in the US and Canada. We're here to share knowledge, consider important questions, and foster collaboration and communication as we strive to better understand and conserve fireflies. This symposium arose out of smaller virtual working group meetings that have taken place annually for several years and that were facilitated by the Xerces Firefly team. These were region-based with meetings for the Southeast, the Mid-Atlantic, and the West. And as time passed, we realized that there was broad interest in many of the topics that we were presenting about and discussing, and also that there was great work happening in places that didn't fit neatly into the working group regions. So that's a little bit of the origin story of this Firefly Symposium, virtual Firefly Symposium. There are other Firefly conferences that happen, such as the International Firefly Symposium that happens every 3 years, organized by Fireflyers International Network. But this week's symposium is all virtual and really focused on the US and Canada. The theme of today's presentations is research and community science. We'll hear from presenters in Utah, Quebec, Massachusetts, Arizona, Pennsylvania, and Tennessee. Like yesterday, there will be 6 presentations, followed by a 20-minute Zoom meeting where there'll be a chance for round-robin introductions and perhaps further conversation of today's topics if time allows. We realize that 3 hours is a very long time to be on Zoom. So there'll be 2 breaks for you to stretch your legs, grab some food, and step away from screens. Amber is periodically putting the symposium schedule into the chat on the events landing page. So you can check the timing of the various presentations and activities today. Note that the times are listed in Eastern time. So I apologize, but if you are in another time zone, you'll need to do a little bit of arithmetic. I just want to, before we start off with our first presenter, I do want to say a big thank you to all of our panelists today and throughout this whole week who have taken the time to put these presentations together and share them with you all. It's it's a great gift to this whole community. I think without further ado, let's kick off. Our first presenter is Christy Bills, Invertebrate Collections Manager at the Natural History Museum of Utah.
All right, so as Richard said, my name is Christy Bills, and I'm actually the entomologist, but my title is Invertebrates Collections Manager here in Salt Lake City. And this is my little talk slide that is a shout out to Chapel Roan. If anybody knows that song, Hot to Glow. It took me a long time to line up all those little fireflies. So I hope you admire it. I work at the Natural History Museum. Could you switch the slide? So I work at the Natural History Museum in Salt Lake City. I'm a collections manager, so I'm not an ecologist, I'm not a taxonomist. I just manage, it's a small collection, it's like 500,000 specimens. Okay, we can. About 14 years ago, 2014, we started a citizen science project to track bioluminescent fireflies in Utah, primarily is how we started. And this came out of a conversation I had with an entomologist at BYU, which is also has an insect collection about 100 miles south of Salt Lake. And he works on fireflies, Seth Bybee, and we decided that we should ask members of the public if they knew where there were bioluminescent fireflies in Utah. So we started this project because BYU had different strengths than my museum. I have like a more robust marketing department and they have more taxonomists. So we thought it would be a good match. And also Utah has kind of like a rivalry between the U and BYU, and being concerned about people's suspicions with academia and land managers and stuff, we thought people would either trust BYU or they would trust me. So we thought we would capitalize on that. And so we sent out these postcards and we sent some emails, and if you can advance the slide. So at the beginning of the project, anecdotally, we knew about 3 populations of fireflies. We were pretty sure just from hearing people's reports, or Dr. Bybee had looked at the fireflies at the south end of Utah Lake, and we knew there were some there, but we thought there might be more if we just ask people. So if you want to switch this slide. This is the reporting form that we use now. When I started this, I did not study any citizen science best practices. I did not know. I had too much faith in the public. And my idea initially was full transparency for everything. So we used a Google form, and then I put points on a map, on a Google map, that were publicly available. And I was warned that there might be problems, and eventually there were. So now we use ArcGIS, and there's more pull down menus. So people don't autofill, like don't type in the text of what they want to tell me. So that stopped people from saying there, when I ask the county, they don't answer USA. So that's kind of like funneled the information to be a little bit more standardized. Okay, next slide. So in all the years we've been doing this, this is some of the data. These are the data points that we've gathered on a map that is not publicly available, but is available to researchers. And then if you switch it again, Richard. We have about 900 data points, and I have combed through them so many times. So when we estimate, we think they're about 95% credible. So like that one near San Francisco, that data point, probably not. Or these ones on the coast of the Northwest, probably not. But we see a lot of reiterations of points at certain localities. So of interest, though, this project has spurred us to change the project from being Utah firefly to Western firefly. So when we started getting reports from Idaho, Wyoming, Nevada, Colorado, we expanded it and developed more partnerships with other organizations so that we could house the information for different states and kind of keep it standard. Okay, next slide. So in the West, we primarily see Pyractomina. There are a few other taxa, but they were seen in the past, and we don't have any recent records. So I don't, and we have been looking. I'm not saying that they're gone, but so far throughout Utah and up into Idaho and Wyoming, partly, we're mostly seeing Pyractomina, probably dispersa. And this is a photo from our collection. We take high res stacked images. Okay, next slide. So we have about 189 specimens, and this map shows where they're collected from. And they are, this information is publicly available on the website Xerces. So you can just search land purity in Utah, or land purity from our museum, or you can search the entire Intermountain West. And if anybody hasn't used Xerces and wants to know how to find specimen records, I'd be happy to show you. I think it's really cool. We can look things up and map it and stuff. And just this recently, we have started doing genetics on the specimens. So we've sequenced 30, and they're in GenBank, and we have them sequenced from 5 Western states. And I'm happy to also send people links so that you can find the exact records, because I find GenBank a little bit hard to search. Yeah, we don't have any like definitive answers yet about like what these things mean that we have sequenced. Like there's not a lot to match it to, or maybe I lack the skills to know how to match it to other things. But we are creating that footprint. We're creating that record that other people can use. Okay, slide 10. So this project's been running a long time, and I have mostly been doing the same things, talking to people, in libraries, nature centers, museums throughout the West. And it kind of expanded this last year where I got to give a talk in Missoula at the insectarium up there, which is super cute if you get a chance. And just as they were about to clip my mic on for the event, they clipped another mic on and said, "Oh, and then we'll also be recording this for public television." I was like, "Oh, that's not what I anticipated." So it's available on YouTube. And if we could switch it again. Yeah, Bugs and Brews, that's right. Okay. We also had a great Audubon article. I don't know, you probably all saw it, but I got interviewed, and I think Candace and a few other people. So Audubon has such a big reach. It was a lot. It felt like we were hitting a lot more audience than just the people I talked to in libraries and on other public radio and stuff. And if we could switch it one more time. Another update from 2025 is that I've been building relationships over the years, but I have a really good relationship with 3 different site managers that want to do some kind of survey or abundance testing. And we see that as the next direction this project is going to go, is like we kind of know where most things are, but we want to do deeper, longer surveys. So we'll be developing some kind of plan for 3 different sites in northern and central Utah, and then a high altitude one near Park City. The most exciting thing that happened for me this year, though, is when I was in Cortez, Colorado, which is the western border, kind of, and it's in the far south and in the west. I was following up on some reports that I'd gotten from a little town called Fruta, which apparently everybody knows, and was, oh, can you switch it one more time? We should be on number 12. So I was in this little town, and I was following up on a report, and all the fireflies I've seen in the last decade in the West start about 9:45, and they're in wet, marshy places. So it could be a field with cows and horses even. It could be just a wetland, but I know what it looks like. So I pulled into Fruta, and it was like this Mayberry charming town. It was so perfect. It was like dusk, and people were riding their bikes, and there was a concert in the park, and there was an ice cream truck, and I was like, this place is amazing. So I was driving up and down all the streets looking for the right habitat, and I didn't see anything. And out of frustration, we just got out of our car and decided to go for a walk and see this cute place. And then fireflies started flashing in the main street on turf, which I have never seen in the West before. It was so stunning. And these lovely people walking by with their dogs and everything are like, oh yeah, we know there's fireflies here. So we went off on a journey with with different people to see where they had seen them throughout the neighborhood, and we saw them in other places in this neighborhood. And they stopped flashing at 9:30, and the people said that that was what they knew to be happening always. So I was completely shocked, and I was only able to pick up one specimen, and there's my crappy little picture of it in a vial. It's really hard to catch a firefly flash. So anyway, it's not great, but we brought it back, and we've been looking at it really closely, and we're going to run a sequence on it, but we think it's Pyralis. So that's really far from its known range, as far as I know. Maybe other people know differently, but it's like the other side of the state. So could be super exciting. Okay, next slide. It's the bad news slide. Our website glitched, so we only got about 49 sightings this year. And Richard, if you could advance it one more time. One population that is not Pyractomina in the southwest corner of the state, we last collected in 2016, and it's we can't find it anymore. So I've been searching every year. I have an entomologist friend who lives down there. I've sent other entomologists down, and we haven't gotten any more reports. So it's an area affected by drought, and we don't know if they're just gone. I don't think it's from growth, because the areas where we find them in Moab have been not built on. So I don't know what's going on, but they seem to be extirpated from that county, maybe. And the other bad news is that my feeling about this project initially was that citizen science projects should give back, and when people report things to me, I'm just going to report them out. But we are getting reports of people being very negatively affected by firefly tourists in their towns. These are really small towns. They're not built for managing anything like this. And inevitably, a social media person snatched our map off our website and was offering it to people with clicks and likes. Like, if you type Firefly in the comments, I'll send you this map. And I was like, that's not your map. Why are you sending that out to people? And I contacted them and they were like, yeah, we can do whatever we want. That my marketing department was like, yeah, they can do whatever they want. But they weren't saying anything like where the map came from, or don't shine lights, or don't collect, or respect private property, which is kind of like the three things I try and tell people. Like, please don't collect fireflies in the West. Please don't, shine bright lights on them. But some neighborhoods are getting so much traffic that they want to sell their house. I've heard they don't even want to be by the fireflies. So we have a really interesting problem in Utah where there are people who are very fervent about fireflies and want to go find them, and there are people who want to protect them, but there are people who are so anti-protecting the sites that there has been a lot of aggression. So there's like a lot of high intensity feelings about this, and I've kind of stepped into it. I didn't know that this would be so adversarial, and we've had to protect sites. So if you can switch it one more time to slide 15, that's the map we currently offer. It's a very uninspiring heat map. You can't even see some of the points in the other states, but we've just found that we can't have people, we don't want to betray is a strong word, but betray the trust of people that have provided data to us, especially on their little farms and ranches where they don't want 15 people lining up on a summer night. Okay, next slide. So this is the slide I usually show when I talk to members of the public. I say this, you can see them from late May to early July, but you can't shop the wild, so there's no place that you can guarantee seeing them. They come out about 9:45 in areas that are marshy and don't have much light. And that's kind of how they behave here in the West that we have seen. So none of this August business that you guys back east get to see. And then another slide, if we can go to the next one, this is kind of how I end most of my talks with the public. So what does it matter? Somebody asked yesterday if fireflies are a keystone species, and I don't know, and I don't think they are, but after I give my talk and people listened, I say, what does it matter? Why does it matter if they are there or if we preserve them? And if we can go to the next slide. This is the point that I end on. Since in Utah and in the Intermountain West, we have such a problem with drought and preserving wetlands and getting people to understand the importance of water in the environment and conservation of land and water. I use this slide about can wonders save us? Like, there's so much fervency to see the fireflies and then sometimes to protect them that I'm hoping that this project will help people want to preserve nearby wetlands because they want to see fireflies, and they don't need to understand the deeper issues about air quality and migrating birds and everything. They're just like, I just want to see fireflies. And so if that gorgeous species or that gorgeous taxa that makes everybody so excited can help preserve wetlands, then they're doing their job, right? Okay, and so last slide. Yeah, I just want to thank everybody for their support. This is really not just a community science project where we have people, but it really relies on a lot of partnerships throughout the West. We're so spread out. There's so much land and many hundreds of miles between us that I can't get everywhere in the short adult season. So I really rely on people in Montana and Southern Utah and Colorado and Wyoming to go fetch specimens or to find them nearby. And it's just been a beautiful collaboration. So that's it. I can take questions if we have time.
We do have about 4 or 5 minutes for questions. So Candice, do you mind opening up the Q&A?
I'm on it. All right, the first one is kind of a comment question, Christy. Someone said that this is a truly wonderful intro slide, the one with all of your fireflies on it. I would be grateful if you and other creative presenters would be open to sharing your slide files. I
For sure. Yeah.
completely lack creative skills. Okay. Yeah, so maybe, yeah, we'll be able to send them out afterwards to attendees. We can chat about that later. Okay, the next question, how do you convince people to care about firefly loss if they've never experienced those populations?
I mean, I can't. I mean, people come to my talks, and I would say 99% of people don't believe we have fireflies in Utah, even though I've been doing this for a decade and I've been on TV, radio, newspaper, I've been at the little libraries, I've been everywhere, but people don't know. And I think what I try and tell people is they say, like, where can I see them? Where can I see them? And I'm very selective now about where I tell people they can go so that it's a protected place where they are publicly accessible. But this is also what I tell people. A lot of times I think in like states like Utah, where we have like a very outdoor culture, hiking, biking, things like that, people think they know everything. They think we've seen everything. There's no surprises. We manage the mule deer. We know how many bears are out there. But I say, like, if we've been doing this for 10 years and I can't find any record of anybody mentioning fireflies in any historical newspaper, like people knew they were here, but like what else is out there? Like we can fall back in love with where we are. So maybe you don't get to see a firefly, but if I'm telling you that there's fireflies in 27 of the 29 counties of Utah, we're not looking hard enough to see cool stuff. So we should do more. We can do more. There's a lot more exciting things to discover. DNA barcode GenBank. I have not talked to them, but I'm happy to send links to anybody because I have links to each thing that we've sequenced, or I can share a spreadsheet with you, and I would love to have anybody else look at it. No, Ben, the genus we find in Utah is Pyractomina, primarily. We see Pyractomina.
But you did see Photuris historically.
Yeah.
Yeah.
Yeah, but those sites are either overbuilt or drought or something. We're just not getting any more reports from those. Yeah. Is that it?
I think so. Oh yeah, Ben. Yeah.
It was probably that Moab specimen from the far southwest. Trying to spearhead the collection of proving difficult. Oh, successful strategy. It's changed over the years. So at the beginning, we sent out postcards to state parks and nature preserves and even stores that sold outdoor equipment. And then we used targeted Facebook ads. So if I knew there was some in a certain county and I wanted to get reports, we would target ads. We don't really try that with Facebook anymore. And we tried doing press releases to rural newspapers because when you're in a small town, your newspaper's, where you get your info. So we tried that, but mostly people find it by Googling. So we have now started asking people where they're seeing the information or where they heard about the project. And a lot of them just say Google. Some of them say the news though, or someplace where I've given a talk. Ben is asking again about the Photuris. Ben is really interested in this Photuris. Let me look up. I will send you a link to the specimens that we found in Moab. So Moab's in the southeast corner of Utah. Super popular tourist site, but like people who live there have like a deep knowledge of the landscape. And there were like 4 or 5 populations very well known in Moab of they're flying higher than the ones that we see in the rest of the state. So I think they are for tourists. And yeah, I'm happy to share the information about it.
Thanks for the questions, everyone.
Yeah.
We're out of time for questions, but I encourage you to reach out to Christy via the Western Firefly Project, or you can also write to Firefly Atlas at xerces.org and we can connect you. And Christy, thank you so much. Feel free to go off video now. Our next speaker, our next presenter is Livia Oliveira, who is a PhD student at Concordia University in Montreal, Canada. Livia, you should have slide control now, so feel free to advance. The next slide should start yours, and take it away. Thank you so much.
Thank you, Richard. As he said, my name is Livia, and my supervisor is Dr. Emma Deplan. I study at Concordia University. And to start, all my background was in plants, so I study mostly population communities of plants and taxonomy, and shifted to a little bit of ecology when I was in my master's. So when I joined the PhD, I had a lot of effort but my supervisor put a lot of faith on me because I came from Brazil, was a new topic for me to study insects. So then all my projects are about charismatic and that can act as umbrella species of insects. And that's how I got into studying how urban habitats management affects plants communities and therefore insect communities. And with this kind of storytelling for my PhD research, I started doing a community science project that was using fireflies to connect science and the public. Here were some photos when I did some research fields classes with my students in Brazil, and some of the species that I found of butterflies here in Montreal for my research already in my PhD. And I don't know if I can play it. Okay, I cannot play the video, but was some videos of fireflies here in the area of Montreal. And then my project was basically based on urbanization is expanding, and this is actually targeting a lot of species and mainly night species that needs that environment more protected to actually thrive in the urban spaces. And insects are very essential for either for habitats, environment, and for us as well, because they act as pollinators, decomposers. They build the whole trophic level they are herbivores, they are carnivores, they are prey, they build the baseline for the trophic webs. And fireflies can act as bioindicators because they are very sensitive to habitat quality. So the goal for this project with fireflies was to track biodiversity and reconnect people with nature. So basically how it works was anyone in the city can participate, reporting sights and absences of fireflies within the city at any night walk time that they had. From June to August is a little different from the last panelist. She was saying from May to early July. I think here in Montreal would be from June to August, mainly in July, after 8:30, 9 p.m., when fireflies are most active. So I built a form that is bilingual because Montreal has two main languages for the whole Quebec. It's French and English. So my form would gather the date and time, the weather temperature, firefly activity with presence absence, the habitat type, location, and other environment features that can connect presence and absence of fireflies, like canopy cover, grass height, artificial light, organic matter, and open water. And I got many of my observations through a lot of help of the Finding Urban Nature, or as we call FUN group, because it's a connection between my university, Concordia, and an organization called Urban Nature, and we're doing night walks for bats and fireflies throughout the whole summer. The first one being late June, but official one in July until October. And this is the QR code for their Instagram account. And For my data summary for my pilot season was in 2024. I got almost 100, I think was 98 observations. And this is the map for my observations in Montreal. For 2025, we got about the same number of observations, but for me it was a little better because we got more spread out across Montreal. We got the north of Montreal that we didn't last time, and more in the west as well. And some basic data that I still need to do proper organization later was firefly activity. Most of the people got presence of fireflies, and not a lot got absences. So this might be a bias for the data analysis. A lot of the firefly presence was within artificial light. Also a lot of organic matter presence when we had fireflies. The land use, this was very exciting because a lot of the population, the community across Montreal got very excited, and they were reporting many of the observations were inside the residential yard, either backyard, front yards. So we have a lot of engaged community here. So this is really nice. And the last one, we were trying to track open waters like lake or something. And a lot of the presence of fireflies was not related to open waters. Vegetation structure, we saw that many of the observations were with a little bit of grass, probably mold, and some of them, of the people that did fill up the form, went to more nature areas with grass that is not actually being mowed. And many of the observations were within low and medium canopy cover. We did some logistic regression with those data points, and we kept only the most important features, that was artificial light, land use, and organic matter. And what we found from these 2 years was that fireflies was strongly negative affected by artificial light. From our observations, we had the highest firefly probability within residential yards, and organic matter was not actually significant for the model, but we saw a slight positive trend to having organic matter for the presence of fireflies. And I think what I got the most excited with was the community observations, because I put a note area in the form where they could say anything they want. So analyzing those, we got that most of the fireflies were seen in gardens, hedges, and low mown yards. Manicured or recent mowed lawns had few or none. Leaf litter, mulch, and dead wood was often present when firefly occurs, so basically organic matter. As I said, light pollution was affecting highly the firefly presence, and they were concentrated in dark shaded areas behind shrubs and fences. And they also saw more fireflies after the rain and alongside edges and near water, ponds, creeks, and bird baths. And as I expected, I'm having a little trouble to pass the slides. Just a second. Okay. As I expected, the peak of observation was around 9 and 10 p.m. from late June to July. And they noticed fewer fireflies than last year. So this is another trend that we can gather having a multiple year project going on, because we can have trends for each year. And also that they were actually enjoying doing the project, because they described the firefly sightings as magical, or the relative for French, féerique. And the feedback of the participants that we had in presence in the night walks led me to add new questions to the forms to capture new features of the habitats. And what's next is we want to test correlation between the firefly presence and landscape metrics like land cover, vegetation cover, or NDVI. We also want to compare firefly presence and absence areas when actually measuring the light pollution. We also want to continue, at least what I'm thinking is until the end of my PhD, the 4 years, monitoring the presence absence of fireflies. And to improve data quality, we were thinking going to the places where the community already see fireflies, because we don't have a good abundance and richness data with my form, but we have presence absence. And with this, I'm sure that if they put, I saw a firefly, they saw a firefly there. So what we aim to do is actually going to the places where they said they had fireflies to try to get species and abundance for the data, and also improve the actual data entry in the form to not having a lot of problems getting the places and other information that they put on the form. And that's for me. Here's a QR code for the website where I explain the form, and I put the link to the form as well. And thank you very much.
Thank you so much, Olivia. We have a good amount of time for questions for Olivia. So folks, if you want to put your questions in the chat, go ahead.
I have a question initially. Have you all considered including like pesticide use or like pesticide practices in your forms when you're asking folks about things like light pollution?
I wasn't thinking of putting pesticide use because, if I'm not mistaken, Montreal banned pesticide use, most of the pesticide use within like residential areas.
Oh.
So if someone put that they use pesticide in their yards or something like this would be kind of against the law.
Oh, interesting.
So I think would not be a good use, but they might use other things, and this can be interesting. So it's something that I can look for.
Okay. Okay, I might go ahead and read these because I'm not sure if everyone can see the Q&A, but someone says, great presentation. I went to the University of Montreal and I'm noticing a lot of observations in the Guy Concordia, Atwater, St. Henri area. Are there metrics in your studies that account for localized participation in the study? I'm just assuming a good number of Concordia students were participating given the usage of the fun group.
Yes, we have most of the observation around one area of Montreal that is mostly the west with the Notre Dame de Grâce neighborhood. But we also try to do more fun activities as the night walks in other places, one more to the east, one out to the north, and one way west. So we try to have different places because from 2024, most of the night walks were around the same neighborhood. But this time we try to get the night walks across Montreal to actually get more information across landscape and not just around one neighborhood.
Great, thank you. Someone asked a follow-up about pesticides, asking if herbicides are used.
So that's the thing. I saw the bylaw announcement. So there are some exceptions, but I don't know which ones. So that's why I said that I can look into it, because might be something interesting about the exceptions that I didn't went through. But if I thought, at least from the bylaw, that they're not supposed to use pesticides or herbicides. I don't know what is the exceptions that they have.
Yeah, it could be interesting to look into. Okay, someone else asks, do volunteers ask about what they can do to increase fireflies? I'm wondering about lighting changes in particular.
Yeah, a lot of them wants to learn more about fireflies, and they ask different amount of questions about, oh, why do they have lights in this color? Can they have different colors of lights? How can I have fireflies in my yards? What I'm doing to not have fireflies because I had 5 years ago, now I don't have it. So it's a good exchange between the community and me and the other researchers, because we can actually we see that the community are engaged and they actually want to learn and they want the fireflies there. So it's a good exchange to explain how to manage the yards, how to for actually having fireflies here next year and everything. So it's really, really nice.
Yeah. Do you all send out updates each year, kind of like a summary of what you found and those recommendations to but the volunteers?
I wasn't doing it because we try to make more anonymous, because from the pilot year, we saw that many of them were recording firefly sites in their yards. So if I did a follow-up, I thought I didn't want the email and wanted to be anonymous because I thought they would be like afraid to giving me the information of their houses and anything. So I
Right.
did totally anonymous. But one thing that I want to do for next year is putting another question saying, if you want to do something more and you want a follow-up, you can leave your email, then I can actually share with them the results, and maybe having them to fill out a more proper form for firefly sightings as the Atlas form that is heavier than mine. But if they are actually interested in doing more sciencey stuff for fireflies, this would be a good measure.
Yeah, that would be great. Okay, it looks like we have another question here. Have any conservation groups proposed reducing light pollution in Montreal?
At least not that I know of.
Any other questions for the Q&A? All right. Feel free to still throw them in the Q&A if they come up later. And yeah, thanks so much, Anna Olivia. That was great.
Thank you very much.
All right, folks, we are at the hour, so we'll go ahead and continue with presentations. Candice, do you want to introduce me, or should I introduce myself?
Yeah, I want to introduce you, even though you got introduced earlier. But yeah, everyone, Richard, he's a conservation biologist here at Xerces and is the Firefly Atlas coordinator and just fireflyer extraordinaire here at Xerces. And I think a lot of you have had the opportunity to interact with him. And I just want to say, Richard and Amber spent a ton of time putting together this webinar series for everybody. So yeah, just really exciting to see all of you here. And I just want to acknowledge all the work that they've put into this. But I will hand it off to Richard to talk about that Firefly Atlas.
Thank you, Candice, and thank you, everyone, really sincerely for being here and for your interest in this topic and in just fireflies generally. Let's kick it off. So 3 years of the Firefly Atlas, stories of fireflies filling gaps in the map. I'm going to give some sort of updates from the Firefly Atlas, kind of where we are so far, but really I want to highlight in this presentation some really neat stories of some of you who have participated and contributed to the Firefly Atlas, who have helped make it better, who have given important feedback, and just made it possible, because it's really all those points on the map are thanks to the efforts and time of people like you. Just a little bit of background with the Xerces Society, in case you weren't here during Candice's talk. We're a science-based nonprofit dedicated to the conservation of invertebrates and their habitats. We're based in the United States and have approximately 100 staff across the country and in Canada. We were founded in 1971 by Dr. Robert Pyle, inspired by the extinction of the Xerces blue butterfly, the first known insect to go extinct in the US due to human causes. And since our founding, we've expanded to work with lots of different taxonomic groups of insects and other invertebrates. We have 5 main program areas: pollinator conservation, endangered species, which is or imperiled species, which is the program that I work within, pesticide reduction, community engagement, and conservation policy. And within all these programs, we use conservation, outreach, applied research, policy, and restoration to advance our goals. I imagine that some of you on this webinar, watching this webinar, are Xerces members yourselves, and I just want to say thank you. Xerces is supported by a really amazing community of members that make our work possible. And if you're interested in becoming a member of the Xerces Society, you can do so on our website. This is a slide that Candice and I show pretty often, looking at kind of the big picture view of our understanding of the status of fireflies in the US and Canada. So in 2019 and 2020, actually before I joined the Xerces Society, the IUCN Firefly Specialist Group of North America, including Xerces, worked to assess the extinction risk of 132 North American firefly species. And we found that a third of these species seem to be doing okay and are categorized as least concern. Think of species like the Big Dipper firefly or the winter firefly. However, there are 20 species that are threatened or nearly threatened with extinction, according to the IUCN Red List criteria. And then notably, over half of the species assessed were too data deficient to draw conclusions, which is not too surprising for insects, but really just shows that we need much more information about our firefly fauna in the US and Canada. And if you project the proportions of those categories onto the data deficient groups, up to a third of firefly species may be at risk of extinction. I get asked many times this summer by reporters, are fireflies declining? Are they in trouble? And I always have to provide the follow-up sort of comment or caveat of which fireflies are you talking about? So firefly conservation is hampered by lack of data. There are many basic questions that we still need to answer about firefly distributions, their phenology, habitat associations, and the threats that they're facing in order to take action that is meaningful and effective. So it was because of these data gaps that the Xerces Society was inspired to create the Firefly Atlas, which is a collaborative community science project to collect data at the species level on firefly species in the US and Canada. And in the Firefly Atlas, there are basically two ways of submitting data. They can blur into each other a little bit, but basically there are incidental observations where, maybe someone found out about the Firefly Atlas after they had seen a firefly or gotten a photo of a firefly. So they submit data that's not as complete as the next category of data, which is species-specific surveys or general inventories, basically full surveys where people go out either looking to see what they find or looking for a particular species of firefly, and then they can report either the firefly observations or it can count as a non-detection survey. Firefly Atlas surveys involve some basic equipment. So it's not just sitting and looking. We do ask and encourage participants to catch fireflies, to get close-up looks at them. We have printable data sheets on our website. And it does help to go out in a group to pool these resources and stay safe while surveying. A complete Firefly Atlas observation would ideally include two main pieces. There would be pictures of the top and bottom of the firefly and flash pattern details. So there are some fireflies where you can know immediately from a photo what it is, and there are some fireflies where the flash pattern is really essential for identifying it to species. So we try and put all these pieces together, and this is sort of the ideal scenario. In reality, we get data that include photos but no flash patterns, flash patterns but no photos, and sometimes we get just reports of presence of fireflies, really more akin to the types of data submissions from projects like the ones we heard about earlier today. Within the Firefly Atlas, when we started this, it was very overwhelming to try and tackle, the 179 species within the US and Canada, and to cover this whole region. So we tried to set some priorities, and we did that using the species that were deemed threatened by the IUCN red listing process, and that seemed like could be documented and verified using a non-lethal community science approach. That said, and you'll see later in the talk, we've gotten some really cool data from outside of these focal regions, and we encourage participation from all across the US and Canada. So before I dive into some of the stories, I just want to throw some numbers out there. We have over 3,000 registered users, which does not mean that they are actively participating. It just means that they created an account and kind of are showing interest in the project. And we've had over the 3 years of the project being active, plus 1 pilot year in 2022. We've had close to 2,000 data submissions, including over 1,200 observations. And within those observations, over 500 could be verified and at the species level. We've had data submissions from 47 states and 9 Canadian provinces. So these are numbers, and they tell a bit of a story, but I really want to get down to places, people, and species to show you just some of the stories that I found inspiring from the past 3 years. So the first species and kind of place I want to highlight is the Florida intertidal firefly, or Micronaspis floridana. It was assessed as endangered on the red list in 2020, and the Xerces Society petitioned this species to be listed on the endangered species in 2023 because of its narrow habitat needs, mangroves and salt marshes, and the numerous threats that it faces, such as sea level rise, coastal squeeze, urbanization, pesticides. The list could go on. So over the past 3 years, surveys done by Firefly Atlas participants and myself have added numerous points to the map, including revisiting sites that didn't have recent records and surveying sites that had never been actively searched. So these are the These are the yellow dots on the map. And I'd really like to highlight the contributions of several Firefly Atlas participants from state and county governments in Florida. So we've had rangers and naturalists from the Florida State Park, from Florida State Parks, as well as naturalists from Pinellas County and Broward County. So the photo in the upper right there is a female Florida intertidal firefly found at a county preserve in county park in Pinellas County, so on the Gulf Coast of Florida. Micronaspis floridana, or the Florida intertidal firefly, is also a good case study for showing the value of Firefly Atlas for getting non-detection data. So this is the not very exciting data points where we go looking for a species and don't find it. This is data that we just don't get from museum collections or sites like iNaturalist that only include observations and not surveys. So this can give us insight into edges of ranges, conditions for detection, and also a better understanding of rarity. On the map, I highlight a little county park in Broward County, Florida, that actually it's a little bit inland, so it's kind of back via the canal system in the sort of Fort Lauderdale area, and it has mangroves, but folks searched the area for Micronaspis and did not find it. Just kind of showing that we can't always use habitat as a proxy for the presence of the fireflies we're interested in. During the Firefly Red List assessments 5 years ago, it was a really common occurrence for the Firefly Specialist Group to encounter species for which the most recently publicly accessible specimen or observation record was decades old. This was the case for a data-deficient firefly called Photinus tenuicinctus, which Dr. Jim Lloyd studied in Arkansas in the early 1960s. You can see a specimen of Jim's here on the right, and you can see on the map that there's just kind of this gray haze of kind of speculation about where this firefly might be occurring today, but no recent definitive data. So 61 years later, master naturalist Pam Morgan, who you'll hear from tomorrow, resurveyed a roadside site in Arkansas and found a thriving population of this species. So revisiting historic localities like this can jumpstart regional survey efforts and put these existing populations back on the radar of researchers and land managers. I also love that Pam came up with an awesome nickname or common name for this species, Ozark Spark. I like this a lot more than what we used in the State of the Fireflies report, which was just the Latin translation, or thinly girdled firefly. Another awesome story is that of Firefly Atlas participant and Xerces ambassador Brandy Dunn. So Brandy Dunn got permission to survey on county conservation land in McHenry County, Illinois, sort of tucked between urban Chicago and the sprawling corn and soybean fields to the west. And Brandy did 4 surveys spread between early June and early August, and she found at least 5 species of firefly. Now, she's outside of our focal regions, but collecting really high quality data. And even for the species that we couldn't get to identify to the species level, there was really nice documentation of the external appearance and the flash pattern. Accidentally skipped that one. I also just want to say kudos to team Bicelonica wickershamorum. So if you look at this pie chart, this is the breakdown of target species of Firefly Atlas submissions. So that blue area is people who just went out and weren't looking for any particular species. But the next largest one was Bisononica wickershamorum as the target species. 184 surveys, or 14% of the total. And Rachel Laura, who's doing her doctoral work on this species, has done a fantastic job of incorporating Firefly Atlas surveys and volunteer participation into her project. Next, I just want to give a shout out to the wildlife agencies at the state level that have participated in the Atlas. Georgia DNR in particular has been really wonderful to work with their access to state parks and other similar state lands and streamlined permitting. They're kind of uniquely positioned to document and really get to know the fireflies of these lands, including some, wow factor, high tourism potential species like the snappy single synchronizing firefly and the blue ghost. But also some species of conservation interest like the loopy 5 firefly and the data deficient Florida sprite firefly. The Georgia DNR team collected this Florida sprite. They collected a specimen, and we actually did DNA barcoding on this specimen as well, which shows that the community science approach and the kind of collecting or kind of more traditional entomology sampling approach can be interwoven as well. So what's next? We just want to continue strengthening the network of participants and providing ongoing training opportunities. Our website is a constant work in progress. And then we're also close to releasing some digital species ID tools for several firefly genera that basically help you narrow down species, but possible species using different criteria, like where you are, what it looks like, the flash pattern in a comprehensive way. So thank you all for your attention. I think I may have run out of time for Q&A, but just want to say I'm incredibly grateful for our funders, the Band Foundation and the De Croizart Fund for supporting the Firefly Atlas, to Xerces members for also supporting our Firefly efforts, and to everyone who has given the Firefly Atlas a chance as we work out wrinkles. So thank you all, and maybe time for one question.
That'll be a very quick question.
Let's see. Someone's asking where they can get a list of firefly species found in their area so they know, so they can get involved in looking for them nearby.
Yep, so on the Firefly Atlas website, we have a filterable checklist. You can use that to get a sort of preliminary species list for your state, and that also includes the red list status as well. So you can get a sense of what might be of more conservation interest. I don't want to take too much time from Cheryl, so let's continue. And please do reach out to me at and Candace at fireflyatlas@xerces.org. Cheryl Mollohan is our next presenter. She's a certified wildlife biologist and independent researcher. Cheryl, you should have slide control now, and we look forward to your talk.
Great. Okay. Thank you, Richard. Can you hear me okay?
Loud and clear. Loud and clear.
Okay. Thank you. So as Richard said, I'm Cheryl Mollohan. I'm an independent researcher. I've worked on fireflies in Arizona since 2019. This is a group project, like most are. Rondé is my field partner, also life partner. Raphael Sarfati is the person who's doing our analysis of our data, and John Howard is the person who actually figured out a way to be able to use trail cameras to monitor firefly populations, to discover and monitor firefly populations. So that's what I'm going to talk about today. So in 2023, we placed 2 modified trail cameras at a site in Arizona, a Nature Conservancy Muleshoe Preserve. This is a site that we've actually worked on since 2021. And it's a known Biselennica wickershamorum site. We call them BWs, also known as the Southwest Spring Firefly. And how we came to have 2 cameras, I was in Ohio in the spring of 2022 talking with another firefly person, John Howard, and mentioned how difficult it is to look for fireflies in desert populations in Arizona. You know, lots of snakes, lots of tough conditions, very hot, often very remote. And the next spring, when I was back, John presented me with 2 cameras. These are cameras that he had been using for monitoring timber rattlesnake dens, and he had messed around with them and thought that they would work for fireflies. So that's how this came about. They were inexpensive Maya-Days trail cameras. We have 3 different models that we've used. They are still on Amazon. And I'll come back to this and provide the specifics. It'll be available in the chat. I mentioned John already. I think he spent a reasonable amount of time messing with this, but he was able to come up with a fix. This is a very easy fix if you know what you're doing, like most things, technology-based. And so this information is also available, will be made available in the chat for you, so you can look at the specifics to this. Okay, so we placed, I actually put out 2 cameras just because we have 2 cameras as our initial try. We had them facing into the site. It really doesn't matter. The question was simple. Can we detect firefly flashes with modified trail cameras? This is BW, or the Southwest Spring Firefly. They're set to come on before sunset, and that's because these guys flash early, in many cases before sunset. The flash was disabled internally. I mentioned that. That's really important. And it was one of the key things was that you were still able to set up a time lapse that continued on after dark. It takes a picture every 10 seconds, so there's no flash, there's no light. It simply takes a picture. It's not based on motion or anything. In our case, they ran for 3 hours this first year to encompass the daily flight times for BWs. As I mentioned, they're early flyers and flashes. So the simple answer to that question is yes. You can see in the circle, this is actually one of our test nights from that night in 2021. And you notice the time, this is before dark. So this was about 15 minutes before sunset. Like I said, these guys are early flashers. So these are 10 seconds apart. So this is just 1 minute, if you will, in what was recorded. So you get slides with, you get slides without, but they're sequenced. Okay, the next set was actually 15 minutes later. The same thing, you can see this is right at dusk. You can see the flashes coming and going. And that's actually one of the things I'll come back to. The fact that the flashes disappear from slide to slide is one of the key ways you figure out that you're looking at fireflies. About now, people always say, well, how do you know for sure you're looking at fireflies? And so I'll go through a couple things quickly. You see the firefly in the yellow circle. Again, these are sequential. And the red circle could certainly be a firefly. And then when you go forward from there, what you can see, the flash is gone. The other light is still there. And if you just watch, you notice that we're advancing. You can advance these quickly. Richard, you can see that, in fact, as he advances, that it doesn't change. And if it was a firefly, obviously it would come and go, and it does not. So Richard, can you advance? Thank you. Yeah, there we go. So the next one, this is a full moonrise. Again, a big cluster of fireflies. This was a super moon, so there was still a lot of firefly activity. Okay, next slide, Richard. Thank you. Another one, and these are some of the trickiest, are night sky, because as you can see with this circle, there's two two dots here that certainly could be fireflies. Okay, Richard, go ahead. They don't move, go ahead. Thank you. And one more time, you notice they have not moved. A firefly shows up, those are stars. And you will also see, so night sky is always a little tricky, you will also see airplanes. But again, it's a movement that you can obviously track, which is not the case with fireflies. Okay, next slide, please. So it worked, our 2023, just using the 8 AA lithium batteries within the cameras. Both cameras worked, both cameras collected firefly data. One lasted for 28 days, the other lasted for 30 days. Next slide, Richard. Thank you. In 2024, I say we loosely because John did this. He came up with an external rechargeable lithium battery and a power cord. Were added to the setup. Okay, go ahead, Richard. We also upgraded to 128 gigabyte memory card. Next slide, Richard. And what we've learned from this, and John had done his research for us, is we could set this camera out and it would last for at least 90 days with this setup, which was amazing. That covers, in most cases for us in Arizona, that covers all of our firefly activity at a given site. Next slide. So our camera results for 2024. Okay, Richard, next one. We put them out just a single night, 9 different times. We actually found 8 new firefly locations. We were working on 2 species, BWs, and we also added in the Western Pyrectomena dispersa in 2024, which was first discovered in Arizona in 2023 by Anna Walker and Raphael Sorpetti. So we were trying to add some sites. And you see, we put them out one night. Lots of times what we would do is we would pick an area, we would pick 2 or 3 sites that looked good, put our cameras out, and then we would still go ahead and do a visual Firefly Atlas survey at one of those close by sites. And it worked really well. It basically let us go from one site to several in a night. Next slide. And we also had a variety of other lengths of deployments. Some of them were just, we stuck a camera up on our way by, we came back several weeks later. It was just very variable. But the bottom line was they were working. We were able to verify these sites as new Firefly sites. Next slide. We did 3 deployments at 3 node sites for BWs that we had already researched earlier that were historic sites for BWs. And the idea of this was to capture the whole 3 months, the entire flash period for that site. And the question being, is this something that we could duplicate year to year and have a trend survey, actually be able to get some information on populations of fireflies year to year. Next site. So this is just an example of the kind of data that we gathered. I figured out the best way to present this was I actually made a little PowerPoint of the information. And this is a new site that we discovered last year for Pyrectomena dispersa in the White Mountains in eastern Arizona. This represents one night. Okay, next slide. So we were at high elevation. So we went from a desert species to a very high elevation species, which was kind of nice after being in 110 degrees in the desert. We were worrying if it was too cold for them to flash. So that was a good switch. Next slide. And so this just gives you again what our setup was. It was 4 hours. Dispersas tend to flash a little later than BWs, so we still had it come on at 7 p.m., but we ran an hour later, so it was 4 hours. And you can see first flash recording 8:11, the last flash was 9:45. 72 flashes recorded over a 94-minute period of time. Next slide. Richard, you'll probably have to start this video. Okay, so you notice, you see fireflies or see flashes of light that you think might be fireflies. I always tell people at this point in time, I've looked at enough of these it's either fireflies or there are little tiny aliens that are invading us in little tiny spaceships, because I can't come up with anything else. This is just a compilation of any picture that had a flash in it. Okay, so that's what I did, and I basically turned it into a video so that we could include it in a way for people to be able to see it. But like I said, the key here is you see those flashes moving around. They're not sedentary. Okay, Richard, you can go on. We don't have to watch all of this. Next one, please. So the question for 2025, you know what, everything worked great in 2024. We felt like we had a very usable technique. The one thing that was missing is identification and flash pattern characteristics. So of course, John went back to work, and go ahead, Richard. And sure enough, oops, something happened. Oh, okay. Yeah, you're okay. So we added a video camera at each of our sites, either 10 or 20-second video. Okay, next slide. Camera was disabled in all the same way, and this is Pyractomina dispersa. Richard, could you go back and see if that one will start? Okay, so this is Pyractomina dispersa. And so a couple different flash patterns, a 3 flash, a 4 flash, and a sputter flash. And you can see all of those are represented here. This was a one-night survey that we did at a news site that we found. Okay, next slide. The next one will be BW. And yeah, and these guys, Rachel had this up yesterday. Single flash, just a second, in between 1 and 2 seconds of flash. In both cases, it worked. I'm comfortable, at least with what I know about these guys, these two species, to be able to identify them to species. Okay, next slide, Richard. So what we've shown you so far was us going out for a night or however many nights and coming back in a darkened room or darkened tent and looking for flashes. And so it worked great for a single night. We would look at when did we start, when was the beginning, what was the end. We had absolutely verified that there were fireflies there, but we felt like there was a lot more we could do with this. Okay, go ahead, Richard. So a long-term deployment, trying to come up with a method to be able to survey sites from year to year, is something in the range of 1 million photos and 21,000 videos. The good news about that is the data is mind-boggling. What is there? You can, on a daily basis, on a seasonal basis, whatever question you want to ask, you can look against temperature, you can look at firefly activity with moon phase. Did you have a monsoon storm? There's just an almost never-ending series of questions you can ask about the data. But in our mind, most importantly, I believe you can compare it both seasonally and annually. So I think you can compare it over time, and that it's a valid way to do that. So in order to have that happen, obviously we needed some additional help. Okay, next slide, please. So this is what Raphael said last year when we gave him the 4.5 million pictures from 2024. So this is a tricky problem because the trail camera pictures have great variability. Daylight, some at night, some with light pollution. All we're trying to do is find tiny and transient specks of light in images that contain many features and bright spots that are not fireflies. And we will get there with persistence and trial and error. Okay, Richard, next slide. And sure enough, a year later, when I asked Raphael to send me some slides, you know, 1 million pictures is not going to be sitting in a tent trying to look for fireflies. Identifying small flashes and compact pictures is hard even for humans. Absolutely, there's a learning curve. Down at the bottom, he feels like he's about 90% accuracy and getting better. And at a cost of about $100, and this has with the 3-month long deployments and getting lower. So we made some great progress this year. Oops, next slide, please, Richard. So I wanted to show you just a little bit of what's possible quickly. So this is just pictures per day, so photos per day. So it was just if there was a photo that had a picture in it, it got counted, and you can see the photos per day, but also his running average, 7-day moving average. This is a BW site in southeastern Arizona, and it really, oops, I'm sorry, Richard, can you go back one slide? Sorry, there we go, thank you. Whoops, okay, one more, forward one more. There we go, thank you. So you notice that, you know, by the time we had the camera deployed in by the 20th, they had already started flashing, but you know, you have a peak sometime about the second week in July, and then you tail off. There's another little peak, but by the end of July, the BWs are done. And this is real typical. It actually looks a lot like what Rachel showed yesterday. Next slide. This, whoops, can you go back one? Thank you. Nope. There's one, there it is. Thank you. So this is another BW site, actually at the same area, same general area. But you notice the pattern is the same for BWs until you get to the end of July, and lo and behold, the flashes continue all the way up into September. And we're pretty sure that this site also has Photinus newly, so 2 species. It was a new site that we identified this year, or last year, I'm sorry. So we're very excited about that. We put up a video camera this year, and the night the video camera was put up, a bear tore it down. And so we do not have our video footage to compare for this year, but hopefully next year. Okay, Richard, thank you. Next slide. We believe this protocol provides a way to survey presence or absence of fireflies in remote habitats over both a nightly and seasonal flight time, and that it can include multiple species that may occur in an area. Okay, next slide, Richard. This particular set of cameras are relatively inexpensive. They run nightly for several hours over a period of months. They worked with video and with still pictures. The good news is you can put them out during the daytime when, if there are snakes about, you can at least see them. And making those hikes in, we put some cameras up in May because we just did not know when the species we were working with started to flash. That was Pyractomina dispersa. But it can run through the whole year. We just last week picked up our cameras that we deployed in June of this year. So it gives you a lot more flexibility of how you use your time in that really crucial time that we all have for a short firefly season. Next slide. This is the trail camera setup, and this one again will be available. You can, and you're always welcome to contact any of us with questions as well. You know, I want to comment on this. There's nothing magic about these cameras. This just happened to be the ones that John was working with. He was happy with them and therefore got passed on to us. Obviously, the expense or lack of expense is a key factor, but you can do this with other cameras. There was another researcher in Arizona last year, Don McClimus. Forgive me if I didn't get that right. Who also came up with they were also successful in using both stills and video. And I think they used Reconyx cameras, if I remember right. So anyway, don't get too hung up on it having to be these particular cameras. If you already have cameras laying around, most of us do, mess with them and see if you can figure out a way to disable them. Next slide. So just some ending thoughts. First of all, field test before you put them out. Stick them out in your yard. John did this to figure out, number one, if you could see flashes on them, but also for the time, how long your batteries are going to work, and so on and so forth. Next one, please. Four-legged site residents are not your friend. Last year, I think we had 3 cameras out of maybe 60 deployments. We had 1 bear. I think we had actually 2 that we had a big water event, 1 bear. And then we had a deer and something we're not sure what it was, coatimundi. This year, things were, for whatever reason, we had more of those. My favorite was, though, we had a deer who came along, turned the camera so that it was staring off into space. And a few days later, a raccoon came along and put it back. So sometimes they can be your friends. This is the reason for the other recommendation is to develop a lockbox or other security system. There's not one for this camera, and this is actually our next step. Because not only do you want to make it so that it is secure, so something can't tear it down, mostly you want it so it doesn't move, because that's where most of the issue comes in. If the bear tears it down, there's not much you can do about it. Video versus still photos, you know, I can talk about this all day, but I think there's value in both. I'm actually a fan of putting both kinds of cameras out there. Next slide. And, you know, work with somebody. Oh, and I missed, yeah, consider duplicate setups. That's one of the reasons. That's what I like about the cheap cameras. Work with an innovative data processor, because you will very quickly become overwhelmed with data. It happens really quickly. And Raphael has been wonderful. The funding, Xerces, thank you so much. Provided some of the funding. Ron and I bought some of our own cameras. And John Howard, I mentioned already. And of course, Raphael is the data analysis person. Next slide. Okay, thank you so much and stay tuned. I'm happy to answer any questions. Please reach out.
Thank you. Thank you, Cheryl. We've don't have time for questions. We're going to go into a very brief 5-minute break. Feel free, Cheryl, if you want to answer questions directly in the Q&A, you can do so. All right, we're at 10 till the hour. It's my great privilege to introduce our next presenter. Dr. Sarah Lauer is a professor of biology at Bucknell University in Pennsylvania, and I am super excited to hear about firefly DNA barcoding from Sarah. Take it away, Sarah, and thank you so much for being here.
Thanks, Richard. I first have to thank the organizers for inviting me to speak today. As has become a running theme today, as I'm sure it has yesterday as well, is the importance of collaboration on some of these studies. So I have many collaborators and mentors to thank, as well as folks who supported me in the field. So many, many heartfelt thanks. My training is in evolutionary biology, but as I've gotten more into conservation, the more applied side of what I do, I've discovered more about conservation and what do we conserve. And one thing that I have learned is that a lot of legislation is at the species level, which begs the question, what is a species? There are many different definitions of species. I took a whole class on it in graduate school, but probably the most common definition has to do with lineages, populations of individuals that are reproductively isolated from others. So this individual cannot interbreed with this other individual. They're separate species, or if they do interbreed, then those offspring are not viable. So this theme of reproductive isolation. So in fireflies, how we tell different species are some of the things that we've heard about today, including what that individual that we catch looks like, both its external and internal morphology, like you can look at their genitalia. We also look at things like when they're active in the environment, what time of the evening, or is it day or night, the type of habitat, where they are on the planet, so their range, and their behavior, specifically for nocturnal species looking at flash pattern. And as a person who studies genetics, I also look at traits of things like the amount of DNA that an individual has. So that would be its genome size, or even the sequence of the DNA that it has. And so one of the issues with this is that some of these traits are difficult to diagnose, so it requires extensive training, or they might require special equipment to look at some of these characteristics. And even if you have those things, some of these traits can even be deceptive. And in my experience working on fireflies in Eastern North America, there are several examples. So one of these is in Photuris fireflies. So here I'm showing you a graph where this vertical axis shows the amount of DNA that individual has, its genome size. And on the horizontal axis, I'm showing you where that firefly was collected. The last two letters are the state abbreviation. And the points are colored by the flash pattern. So we had some that were doing a slow blue kind of flash. Some were obviously synchronizing their flashes and were Photuris frontalis. Some were doing a single flash like Photuris heavis, a tremulans-like flash, or even a pyralis-like flash. So what you should notice here is that some of these flash patterns all have a similar genome size. So all the red ones that are Photuris frontalis, those all sort of group together in the amount of DNA they have. And that's expected because these individuals they are one species, they can interbreed. But if you look at the slow blue group next to them, there seem to be some that have really big genome size and some that have a smaller genome size, which suggests that maybe these cannot interbreed and there might be separate species within this. So super confusing, Photuris. This isn't unique to fireflies. It happens in other organismal groups where sometimes traits can be deceptive. So one solution that's been used with success in other organismal groups is DNA barcoding. So this is where you go out and you catch your specimen, you extract its DNA, and you compare the sequence of that DNA to a bunch of sequences of DNA that come from a specimen of known species to say, oh, okay, I have this species because it has the same DNA sequence or a very similar DNA sequence. In practice, this is slightly more complicated. So we go out, we collect our sample. I store my samples in ethanol, among other things, if they're for DNA barcoding, mostly ethanol. Bring them back to the lab and extract DNA from leg tissue. So DNA is made up of building blocks called bases, and they're symbolized by these letters A, T, G, and C. And in fireflies, depending on the species, they may have anywhere from 400 million to 2 billion DNA letters in their genome. Now, it's technologically difficult and super costly at the moment to sequence all of these DNA letters. So for DNA barcoding, we actually, in practice, only sequence a very small segment of that. So only about 700 DNA letters. And that's from a specific gene called cytochrome C oxidase subunit 1, abbreviated as COX1 or CO1. And this particular segment is slightly, or it's very special. So most of the DNA of the firefly is hosted in the nuclear genome. So in the nucleus of the firefly cell, that's where most of those 400 million to 2 billion letters hang out. And they have the instructions, all the DNA instructions to build that particular firefly. But there's also these organelles called mitochondria. This is where a lot of metabolism happens, and they also have their own genome. In fireflies, that genome is about 17,000 DNA letters, and the part that we are sequencing is just this little 700 base pair part, so 1/2 of cytochrome C oxidase subunit when we're talking about DNA barcoding generally. Not all the time, but usually. Mitochondrial DNA is also different from nuclear DNA in the way that it's inherited. So nuclear DNA, you get half, in fireflies, you get half of the genetic information from mom, half from dad. So it offers sort of these two stories of how the DNA is inherited, whereas the mitochondrial DNA is only inherited from mom. So it's actually really difficult to visualize a single DNA molecule. So in practice, after we've extracted the DNA, we make lots and lots of copies of just that little tiny 700 letter CO1 fragment using a technique called polymerase chain reaction, or PCR. We get that sequenced. Here at Bucknell, we use Eurofins as our company. We send out all our sequencing because we're a small institution. When we get the data back, we manually check it for accuracy, and then we compare it with sequences of species that we know their identity. So here I'm lining up the sequence, the CO1 sequence of this question mark species and comparing it with species E, A, B, C, and D. I then use a computer to infer how these individuals are related, and it comes out with this, what we call a phylogenetic tree, or a tree of species relationships. You can think of it sort of like a family tree. So this tree is showing you that my question mark species is closely related to species A, and species B and C are closely related, but then those two groups, question mark A and B and C, share a common ancestor back in time. And then finally share a common ancestor with species E. So in this toy example, my morphology, the color of my sample, matches with the story told by the DNA that it's most closely related to species A. And so in this case, I think that question mark is species A. Awesome, celebrate, party for all. But in practice, it is usually not this simple. So if my slide will go, you run into cases often where the DNA does not match the phenotype. So here,
just interjecting, it looks like it hadn't advanced. So you want to be on the slide with the 3 phylogenetic trees, right?
yes, with all of the exclamation points and angry symbols.
Okay, you're back there. Can you? I won't mess with anything. See if you can advance, and if not, I can.
I appreciate it. Thanks. So, for example, so on the question mark equals species A box, that's the one that we just looked at, but you could run into a situation where there are different individuals that look differently, but they seem to be closely related genetically, or even there's some new morphology that you're not quite sure where that fits in. And this could be because there's population level variation. It could be maybe you've discovered a new species, or maybe CO1, I told you, is only telling part of the story, the genetic story. And so maybe it doesn't have enough information to even tell us what the species is. So I'm going to talk about 3 stories of how I've been using DNA barcoding to look at different groups of fireflies. This first one is in the genus Photuris. So Photuris reticulata, or the blue ghost firefly, is one of my favorite fireflies. They, instead of flashing, they glow. And they have a pretty restricted range here in the Southeast, not as restricted as Photinus floridanus, but they have a fairly restricted range. And when I was in graduate school, I was interested in sequencing their genome to learn more about their really cool bioluminescence. So I went about, I caught individuals near University of Georgia, where I was getting my degree, and got estimates of genome size to know how much do I actually need to sequence to get the genome. And what I found was that I collected individuals in Athens, Georgia, near the University of Georgia, and then about 30 minutes down the road in Watkinsville, Georgia. And when I looked at the genome size estimates, they seemed to have come in 2 flavors. One was had a group about 835 million DNA letters, and the other one was up in the billions of DNA letters. And so to me, this suggested that there was a new species here. When I went back to my specimens and looked at them closer, and I tried to key them out, they actually keyed out not to Photuris reticulata, but to Photuris inextensa, a totally different species. Now, if you're familiar with Photuris inextensa does not have any lanterns. But these specimens that I had, even though they sort of looked like Photuris inextensa everywhere else, they did have lanterns. And so that was another clue. Oh, maybe this is a previously undescribed species. So I DNA barcoded these species, looking at that CO1 segment. And here I'm showing you the tree of species relationships generated from that data. So I got some additional specimens from my own collections, but also from Lynn Faust. Many, many thanks. And what you should notice here is that there's a group of Photuris reticulata that were collected in northern Georgia, Great Smoky Mountains National Park, in Knoxville, Tennessee, and in Athens, Georgia. Those are all closely related. But then there's also those Watkinsville, Georgia specimens, and they are actually most closely related to known Photuris inextensa samples from Tennessee. And they're more closely related to those samples than they are to the Photuris reticulata collected from other places. So the DNA confirmed that morphology story in the genome size data, and now this species has been described and named as Photuris christinae after the first author's mother. I also have used DNA barcoding to look at variation within a species. So the common eastern firefly, Photinus pyralis, is one of the most widespread fireflies in the eastern US. And across its range, it varies in its morphology. So on the east coast, we can get pyralis that look more like the top picture, where there's like a really nice dark dot on the head shield. But if you collect individuals in Texas or other places sort of in that area, they might be missing that dot. They might be a little bit longer. They have a slightly different morphology. And so as part of my PhD work, I went around sampling Photinus pyralis from many different sites, looking in the Midwest and the East Coast and down in Texas. And then I DNA barcoded those samples, and I know the writing is very, very small. But what you should notice from this tree of relationships is that there's one group of really closely related fireflies, or have a very similar DNA barcode, that were all collected on sort of the eastern part of the US, Pennsylvania, Georgia, Tennessee. And then there's this other group of fireflies that is sort of outside of that were all collected in Texas and Arkansas. These are all Photinus pyralis, but they seem to be dividing up into groups geographically. And I followed this up with a study of looking at not just that DNA barcoding segment, not just that CO1 gene, but actually across the genome, teaming up with Anna Catalan. And she was able to identify 3 separate groups of Photinus pyralis within the big species. So there's an eastern group, a central group, and the Texas group. And they seem to be genetically different from one another, with the Texas group actually having the most genetic diversity of any of these Photinus pyralis groups. And so what this suggests actually is if we ever become worried about Photinus pyralis, we want to promote its conservation, we may want to have separate conservation areas or plans for different regions because they represent more special genetic groups that we want to preserve. So my last story has to do with the firefly Pyrrhipiga decipiens. So it's a small firefly. And it's something this is a question I'm still working on. So these are Pyrrhipiga decipiens that we collected in 2012. So with Lynn Faust and Katrine Stenger-Hall and Raphael de Kock in Allegheny National Forest, with Ken and Peggy Butler as well, as part of surveying Allegheny National Forest for species diversity. And again, I was interested in sequencing genomes, and so I got estimates of genome size. And what I found again was that there seemed to be 2 different genome sizes, one that was smaller, about 700 million DNA letters versus the over 1 billion DNA letters. Oh my goodness, another species. But it turns out that there's actually a couple of look-alike species in this area. So one is the target species I thought these all were, Pyrrhipiga decipiens. And another is Pyrrhipiga minuta, which again is in the same geographical location, about the same size. And the diagnostic character to tell these 2 apart is, are they wide or are they slim? And then there's some difference in the stripe on that head shield. And this gets even more complicated because there's another species called Pyrrhipiga nigricans, also in the same area. It's slightly larger, but still overlaps in size with the others. And the distinguishing character is that it has a dark border around its head shield. This is complicated by the fact that Pyrrhipiga decipiens can also have an entire head shield that is dark. So they all look very similar. They're all small. They're all day-active species. You can look at their genitalia and try to discriminate them that way. But when I did these genome size measurements, I hadn't done that yet because I thought that they were all Pyrrhipiga decipiens based on the external morphology. So my hypothesis was that maybe I messed up my ID. So I DNA barcoded these samples. And what I found was that all of the Pyrrhipiga decipiens with a large genome size grouped with the ones that also had a small genome size, but the small genome size were in sort of a group by themselves. So maybe what I did is I messed up and I actually had two different species there. Okay, beginning grad student, my bad. So since then, I have been on a quest to nail down the answer to this question. And we've been collecting at sites across in Pennsylvania and in Vermont, as well as getting some additional samples from Lynn Faust, and getting morphological expertise from Sarah Rivera and Luis Felipe Lima de Silveira in Western Carolina University. And so what we have confirmed is that all of our samples are Pyrrhipiga decipiens based on their genitalia. We've collected a bunch from Pennsylvania and Vermont, including ones that have that dark head shield, and they all seem to be Pyrrhipiga decipiens. What I should point out to you, so this top group is that sort of teal. Those are all from Vermont and Pennsylvania. There's pink that are just from Pennsylvania, from a grassy area. We've got some that we're not exactly sure that they're from. The blue box shows you those original large genome samples, and the small genome samples are most closely related to those. And then two that we collected at the Heart's Content site in Allegheny National Forest group with those two small genome samples. And then all outside of those are Pyrrhipiga minuta from Tennessee. So it seems like, based on the fact that it's unlikely that these small genome and large genome size individuals can interbreed because they have such drastically different amounts of DNA, is that we might actually have multiple new species here. So each color might represent a new species, or it could be that CO1 is just not having the right data to help us distinguish species in this way. So jury is still out. I'm still frustrated, and I hope to know an answer one day. So this isn't just in these three groups. I've told you two stories of success and one that is still frustrating. In Pyractomina, DNA barcoding using CO1, you should use with caution. So there's some data that's up on GenBank from Katrine and me. It shows Pyractomina borealis doesn't work very well with DNA barcoding. That analysis hasn't been published, but the data is there if you want to check it out for yourself. And then if you look within Photuris, Katrine and Jim Lloyd showed that in some groups, you can't actually identify an individual to a specific species within Photuris. So I want to reassure you that there is promise in DNA barcoding. It's super useful to get to genus. It can be useful to get to species. And I think where we're at right now, and I think we heard about this earlier from Christy, is that we need more data to figure out when and why it's not working. So we need more sampling across populations and species, and potentially more sequence data to help us identify maybe a different segment of DNA that could be more useful. So with that, I'm happy to take questions if I have time.
We've gone a little over, Sarah, so if you don't mind answering, you should have access to, if you click on Q&A, you should be able to type answer to people's questions. And that way people can still get those questions answered. Thank you so much. That was a fantastic talk. Our next speaker is Jeff Hill, who is an area interpretive ranger at Tennessee State Parks. Jeff, I'm just gonna give you slide control, and we can make sure it's working, and take it away.
All right, can y'all hear me?
Loud and clear.
Perfect. And slide control works. It looks like there's just a little bit of a lag, and I can deal with that. So my name's Jeff Hill. I work with Tennessee State Parks. I'm a regional support position based out of the middle of Tennessee, but I have access to all of our full-time employees across the entire state, all the way from Memphis, all the way out to the mountains out east, as well as our extensive network of volunteers. And I will explain why that is important in the next couple slides. Today, I wanted to highlight a project that I started that was just kind of by chance. I got lucky and I had a passion, so it worked out. And then a couple projects that we were able to help out on because of that. So I identified pretty quickly when I started interacting with the firefly world, pretty much everybody on this symposium right now, that there's a pretty big gap in firefly research, just like there is in most biological research. When I was getting my bachelor's degree in wildlife biology, the same gap existed. There's institutional research going on. It's very technical. It's usually not super accessible to the general public, based on the level of knowledge that's needed to understand the research, as well as some of the equipment and stuff like that. And then kind of on the opposite end of that spectrum, we have community science. I'm sure you've noticed in the past 10 years or so, iNaturalist and similar apps have exploded. The public has adopted them very willingly. And my goal is kind of to try to bridge that gap where possible, to maximize the efficiency of y'all's projects, no matter where you are studying. So I've been able to get in touch with some researchers. I'd like that number to increase. That's kind of why I'm talking here today, and connect them not only with the resources that my agency promotes and protects across the state of Tennessee, but also the people. So whether this is full-time staff, part-time staff, or volunteers, we have quite a few people who are pretty highly skilled, whether it's just a hobby that they really took to, or they have some traditional training with a biology degree and they're just pursuing a career elsewhere. And this is kind of how they can help out with science still. So the first one, this is my project. And like I said, I'm not nearly as smart as y'all. I can't come up with projects on my own. This one just fell in my lap. It was a project studying the habits of walldoxeyi larvae, specifically in West Tennessee. As part of my job at the time, I was a park ranger, and I spent most of my days leading canoe tours in a cypress swamp. And we were out on a evening cypress canoe paddle that went a little bit long. It went well into the night, actually, because I was enjoying it. And one of the guests on this paddle back in October of 23 said, "Hey, what's that glowing or flashing at the bottom of that cypress tree?" And I had no idea. So I paddled over, and it took me several minutes to relocate this flash. And it turns out that it was a tiny little firefly larva that y'all probably would recognize right away. But me, being a general park ranger, wildlife biology degree, I had a hunch of what it was, but I wasn't really sure. So I got some pictures of it, jotted down some key details, and just like any inquisitive person, I went to Google and I quickly found Lynn's email and shot her an email with a couple pictures, introduced myself, and said, "Hey, is this interesting?" I honestly didn't expect a reply. I owned her field guide. I know I'd promoted it to dozens and dozens of guests as kind of a really good starting point for getting into fireflies. And she replied, I think that same night. And she said, wow, it's definitely larva. And based on the habitat you just described, I'm very suspicious that it might be walldoxeyi larva. At the time, there was only a handful of known records of their larvae. So there was some significance on this observation, and she kind of challenged me, hey, can you go find some more? At my park, I was able to find some more the following night, and I kind of got a general pattern of what was going on. They tended to inhabit the bottom 6 inches or so above the water line. And I had a network of volunteers that I was able to get in touch with to survey half the state over the course of about a month. But from that first night to the first survey that I wasn't doing, but the team was doing, it was about a week. So I was able to identify, give some basic training, a basic guideline on how the sampling should look. And then we were able to enact that study really, really quickly across the state and get that data back to Lynn. And I think that's why she put me in contact with Richard and said, hey, Jeff might be on something here. Not that he's going to go get a master's degree or PhD, but he's able to mobilize people very efficiently across the state of Tennessee. So that went by. We were really excited. And then I got an email from a student out of University of Colorado. I might be getting ahead of myself here. Sorry, getting ahead of myself. So Owen Latham out of University of Colorado, he was doing a survey. He reached out to me. He had heard my name through Lynn and Richard, I believe, and said, hey, would you be willing to find somebody to do this? Very quickly, he was able to ship us the equipment. Owen's project, very similar to the trail camera project. It was really cost effective to send those units across the country. There's always some inherent risk of damage during shipment or somebody walking away with your equipment that you don't have your hands and eyes on. But at the cost of $100 for a trail camera, where Owen was using some GoPros at the time, I think the cost was less than a couple $100. So we were able to gather data from him for his project in Colorado, all the way out in West Tennessee without a problem. And then this spring, I was contacted by another student, Noel Clark out of University of Georgia, who was looking for some blue ghost samples. It was a simple, hey, I'm looking to collect, there was a couple species Noel was looking for. Do you know anybody who A, has these present in their area, and B, would be able to collect them for me and send them back? And it was as easy as I directed Noel to our permit, our scientific permit link on our agency's website. Once she got that approved, I was able to reach out to a team. Unfortunately, it was kind of last minute. We'll plan better for next year. But we were able to get one volunteer in Middle Tennessee to collect 3 specimens, I think, which doesn't sound like that much, but those 3 specimens were produced by just Noel sending an email. So the moral of the story is, if you're interested in increasing your sample size, specifically across the state of Tennessee, if we contain your study species, feel free to reach out to me, and hopefully I can get in touch with some volunteers or some field staff, and we can help maximize your sample size as pain-free as possible. Yeah, that was super quick. I just wanted, really, when Richard said, hey, do you mind talking? I said, yeah, I would love to promote some more Tennessee biology. So if you want to reach out to me, my contact is on the screen. Richard has my contact as well. I'm sure you could find it somewhere on the Symposium's website. If you have any questions or anything like that, I can hop on the Q&A, but that's all I got for you.
Thank you. Thank you so much, Jeff. I think your talk really showed the power of collaboration across different sectors within the Firefly community and the value of just continuing to grow the Fireflyer community. So thank you. Thank you so much. And let's open up the Q&A to see what questions we have.
Read them off. So the first question that we have for you, Jeff, do you find that you see more than one species when adult males are flashing in the cypress swamp?
Do I see more than one species? Yes. Typically, it's uncommon for us to go into a study site and it exclusively be walldoxeyi.
Great. Oops. We have another question. How do you recruit volunteers for this kind of survey?
So we have, and this probably doesn't apply to you, Zach, but we have a robust volunteer program within Tennessee State Parks. We actually have a specific one that we call a regular service volunteer, in which essentially we could put out a job listing for a volunteer. So there could be required skills, required experience. A lot of times with those, we are finding people who have biology degrees or maybe a retired biology teacher or something like that, who's just looking for a way to contribute towards y'all's studies. And besides that, it's really just keeping a large professional social network. I know Corey's on here, me and Corey, who provided all the photos, the backgrounds of my photos. He was one of the first people I reached out to when Noel reached out, because I know he's very passionate about firefly education with the public, as well as photographing them, obviously.
Great, thank you. Let's see. Have you seen tree preferences in larvae in terms of DBH?
Great question, Grace. Not so much about the diameter preference. It's really some, and I'm not a botanist or dendrologist, but some cypress trees in standing water will have very elaborate buttresses with lots of folds and crevices, and some will be just a cylindrical bowl or trunk of the tree. The ones that have the buttresses with lots of folds and creases typically were the ones we detected the larva on, even though they had more hiding spots. The smooth trunks tended to not have any larva on them. And once again, this was all within about 8 to 10 inches of the water line. It's where the capillary action kind of wicks up some of that water. So during the daytime, you can actually see that dark band right above the water. That's almost exclusively where all the larvae were.
I think that might be all. Any other questions?
Thank you so much for having me. If anybody else has any questions they think of later, feel free to send me an email. Whenever I get a random Firefly email during my week, it always brightens my week. So thank you very much.
Thanks, Jeff. I just saw this other question from Grace. Do we want to do this one really quickly?
Yeah, go ahead.
Okay, one more for you, Jeff. Have you found that these larvae are in places that floods all the time versus seasonally?
Yes, great question. The ones that we detected were in permanent bodies of water. We did survey in a couple vernal pools that contain cypress, and we did not find any larvae there. Our sample size was pretty small, and it was only over the course of 1 month. So take that with a grain of salt, but yeah, standing water, permanent water.
Great, thank you. Okay, now you can take it away, Richard.
That was our last presentation of the day. Thank you all for your participation, great questions, your attention. Tomorrow will be our third and final day of the symposium, starting at the same time and using the same Zoom link for the webinar. Tomorrow's theme is really about sharing fireflies with other people. So viewing events, education, and firefly tourism. I'm really excited for this set of presentations. So please join tomorrow. Thank you so much, and yeah, appreciate your attendance.